In protein synthesis, there are two parts to this, two stages to this. And the first one is called transcription. The transcription occurs in the nucleus, and it's where we get the gene. So the gene from the DNA is transcribed onto mRNA. So we're getting our recipe for our protein. So in transcription, we have our DNA here. This is DNA. And this is a section of DNA that contains that gene or the recipe. And what we do is we rip it apart. So separate the strand, and that is done through an enzyme. And the enzyme is called the promoter. So the promoter separates the DNA strands. So it uncoils it and then unzips it to expose the two templates. So this side is a template, and this side is a template. And either side can be used, okay? So if we use this side down here, if we use that side there, we have an enzyme, and this enzyme is called Rna Polymerase. And we attach nucleotides. Let's use pink here, it's fine. So we attach the nucleotides here. We add nucleotides, but they're not actually attached. We match them up, and then once they're matched up with the template here, then we separate it or detach it from the DNA. So we add the nucleotides there, and then when finished, then when we're done, then the mRNA detaches. From the DNA. And then the DNA comes back together and coils back up and is back to the way it was. So it zips back up. Now keep in mind and remember that this is RNA, so those nucleotides are C to G and A to U. So this is where we start to use the U. So wherever there's a T in here, so if this is a T, then this one here is going to be a U. Or sorry. So if this is an A, this is a U, sorry, yeah. Okay, so if this is an A, then this becomes a U. So it detaches, and then what happens to it? Well, protein synthesis occurs in the cytoplasm. So the mRNA goes out of the nucleus, so leaves the nucleus into the cytoplasm. And that's all there is to transcription, okay? So once we have the mRNA and it goes out into the cytoplasm, then we make the protein and we enter the next stage.